Hello everyone, welcome back to my channel. I hope you all are preparing pretty well for your upcoming examination, that is DBT-JRF, and you have already watched the previous session. If you have not watched, I will provide the link of those videos in this video. So this is the first question. So let's solve the very first question. You can easily get 3 marks from this question. You have to match column 1 with column 2. So you can see here different processes are mentioned in the column 1. The first one is caramelization, denaturation, oxidation and bleaching. So इसका आंसर कैसे निकालना है? अगर आपको सारा कुछ पता नहीं भी है then also you can answer this question. अगर आपको सारा कुछ नहीं भी पता है so then also you can answer this question. You know that caramelization of sugar takes place, यानी कि P का 2, तो P का 2 किस में है? P का 2 है फर्स्ट ऑप्शन में एंड सेकेंड ऑप्शन में, तो डोंट लुक फॉर द थर्ड एंड फोर्थ ऑप्शन. After that, you know that enzymes are proteinaceous body and that are destroyed in harsh environmental condition. Like if you alter the pH, if you raise the temperature, the protein will be denatured. So the denaturation takes place in the enzyme, यानी Q का 4. So Q का 4 किस में है? सिर्फ फर्स्ट में, यानी कि ये भी एलिमिनेट हो गया ना. ऑक्सीडेशन, so oxidation of fat or lipid occur, which is also known as rancidity. And bleaching takes place in the pigments. So you can see how easily you got 3 marks by answering in such a way that आपने बिना ही चारों ऑप्शन को जाने आपने आंसर को पिक आउट कर लिया थ्रू योर स्मार्ट स्ट्रैटेजी. सो यू हैव टू अप्लाई this smart strategy while solving each and every question in the paper, बिकॉज सब कुछ सबको नहीं पता होता है. सो आपको वो चूज करना है जो आपको पता हो. कैरमलाइजेशन इज द वेरी इंपॉर्टेंट टर्म, वेरी कॉमनली यूज टर्म इन केस ऑफ शुगर. सो उसके थ्रू भी आपने 50% ऑप्शन तो वहीं एलिमिनेट कर दिए थे. नाउ डीनेचुरेशन ऑफ प्रोटीन अकर्स इन हार्श एनवायरमेंटल कंडीशन, सो थ्रू दैट आपने करेक्ट आंसर पिक आउट कर लिया. नाउ लेट्स मूव ऑन टू द नेक्स्ट क्वेश्चन. सो इसका करेक्ट आंसर जो है वो हो जाता है option first. So let's solve this question, which is about the overall shape of a bacterial cell is determined by which of the following: cytoskeleton, cell wall, nucleoid, cell surface membrane. So a bacterial shape is more or less defined by the cell wall present in it, because cell wall is a non-living and very tough coat outside the plasma membrane, so it will decide the shape of the particular bacteria. So the right answer for this question is option B. Now next question: which of the following biomolecules are formed by condensation reaction? Polypeptide, polysaccharide, steroids or nucleic acid? So more than one option can also be correct. So this polypeptide, polysaccharide and nucleic acid are made up by the condensation reaction. So the right answer for this question is A, B and D. So all these three are, this polypeptide, this polysaccharide and nucleic acid are synthesized by the condensation reaction. The correct answer will be A, B and D. Now again one question: condensation reaction can occur between which of the following pair of chemical functional group? In this question also more than one option is correct. So hydroxyl and hydroxyl group, carboxylic and carboxylic group, amino and carboxylic group, carboxylic and hydroxyl group. Please select all that applies. So a condensation reaction can also occur in between an OH and an OH group, a carboxylic and a carboxylic acid group. So दो हाइड्रोक्सिल के बीच में भी बन सकता है कंडेंसेशन रिएक्शन, दो कार्बोक्सिलिक के बीच में भी हो सकता है, एक अमीनो एंड एक कार्बोक्सिल के बीच में भी बन सकता है, एंड एक कार्बोक्सिल एंड एक हाइड्रोक्सिल के बीच में भी बन सकता है. यानी कि ये चारों ही ऑप्शंस क्या हैं? ये करेक्ट हैं. All these four options are correct. So a chemical bond can be formed with the elimination of water molecule. So that is by a condensation reaction between all four listed pairing. यानी जितने भी यहाँ पे चारों ऑप्शन हैं उन सब में ही क्या बन सकता है? कंडेंसेशन रिएक्शन हो सकता है through the elimination of a water molecule from them. So all of the above mentioned options are correct. So now you have to match each type of biological polymer with its appropriate type of residue. So polypeptide is made up of what? Polymers of amino acid. Ribonucleic acid is made up of what? Ribonucleotides. Polysaccharide is made up of so many monosaccharides and disaccharides. Deoxyribonucleic acid, that is DNA, is made up of deoxyribonucleotides. So the correct answer is polypeptides is polymer of amino acid. Ribonucleotide is made up of ribonucleotides. Polysaccharides is made up of monosaccharides, and deoxyribonucleic acid, that is the DNA, is made up of deoxyribonucleotides. So this type of question can be frequently asked in your DBT-JRF examination. So now next question. Within the nucleus, individual chromosomes are thought to occupy discrete territory. Which of the following is most likely to promote this segregation? Nuclear lamina, nuclear pore complex, nuclear matrix scaffold, or intermediate fiber. So we all know within the nucleus the individual chromosome occupy a discrete location. So why this is so? Why this is so that the chromosome occupy a discrete region? So the region for their segregation is nothing but the nuclear matrix scaffold. So the right answer for this question is option C, nuclear matrix scaffold. Now one more question, which is again a matching question. All these are very important. You can make notes, you can just write it down in your copy, what are the crisp points through which you can solve so many other questions as well. So also known as primase generate short RNA primer. So this is related to the DNA replication part. DNA replication का ये match the following है, which is known as the primase. So DNA-G is basically known as the primase, which is responsible for generating or synthesizing short RNA primers to initiate the whole DNA replication process. Synthesizes DNA by moving along the parent strand in the 3' to 5' direction. So it is the DNA polymerase 3 which is responsible for this. Possess 5' to 3' exonuclease activity, which is the activity of DNA polymerase 1, and which is responsible for sealing the nicks in the daughter DNA strand. So it is the DNA ligase which seals the gap by formation of phosphodiester bond. So the correct answer for this question is DNA-G act as a primer, DNA polymerase 3 act as a synthesizer of the DNA in the direction 3' to 5', and who possess the exonuclease activity? DNA polymerase 1 possess the 5' to 3' exonuclease activity. DNA ligase is act as a molecular glue. It seals the nick in the daughter DNA strand. So this is again an important question which can be asked this time also. So these are the pointers. DNA-G is a primase that can generate short sequences of RNA. So this DNA polymerase 3 uses the RNA primer synthesized by this DNA-G to help it to bind to the parental DNA strand, and it is helping to synthesize the new daughter strand moving along the direction 3' to 5'. Now DNA polymerase 1 possess 5' to 3' exonuclease activity. So this activity is responsible for the removal of RNA primers from the replicated daughter strand, and DNA ligase seals nick in the DNA such as those formed within Okazaki fragments during the DNA replication. So all these pointers are very much important. Now one more question can be asked, that is the antibody affinity chromatography. So now one more important concept that can be totally asked in the DBT-JRF examination is this, specifically antibody affinity chromatography. What is antibody affinity chromatography? Affinity chromatography क्या है? इसमें हम लोग क्या यूज करते हैं, स्पेसिफिक मॉलिक्यूल्स यूज करते हैं सो दैट इट विल बाइंड टू द प्रोटीन ऑफ इंटरेस्ट, इट विल जस्ट hold tightly the protein of interest, and all other molecule will be eluted out. So we are using specific molecule so that it will bind with the protein of interest. So यही principle होता है, like in this column you have used antibody as the specific molecule which will bind your protein of interest. So see, in this column you have provided your antibody, so this red protein is to be recognized by this antibody. So this blue protein is the another protein which is present along with your protein of interest. So red one is the protein of interest and this blue one is unwanted protein. So you need to purify this. So you need to purify this red one from the mixture of blue and red one. So what you will do, you will just put some specific antibody that will only bind the red one and not the blue one. So here you can see you have put so many important this antibody in the mixture of the protein in which both blue and red protein are there. So what it will do, it will specifically bind to the protein of interest, that is the red one, and the blue one will be eluted out. So all this blue one are eluted out, and their specific molecule, that is the protein of interest, is binded to the antibody. So after washing it, after altering the pH, you can elute this out, and through that you will get your own protein of interest. So this is how a protein purification can take place. So this is important. The ability of protein to bind specifically to a molecule is the basis of affinity chromatography. affinity chromatography का ही प्रिंसिपल होता है कि कोई भी प्रोटीन जो है वो स्पेसिफिकली जाके किसी एक मॉलिक्यूल से ही बाइंड होता है. So in this technique, ligand molecule that bind to a protein of interest are covalently attached to the beads used to form the column. So covalently attachment हुआ होता है because अगर covalent attachment नहीं है, weak interaction है, so it may fall off. So through that it is purified. So ligand can be enzyme, substrate, or any other small molecule that bind to specific proteins. So this technique is widely known as antibody affinity chromatography. The attached ligand is an antibody specific for the desired protein. जो भी अभी मैंने explain किया उसे कहा जाता है antibody affinity chromatography, because you are using an antibody to specifically bind to your protein of interest to purify the mixture of protein. For an app, so solve this question. For an application where you require a sample of your target protein at high purity, what would be good purification strategy? Assume that your starting point is E. coli cells in which the target protein fused to an affinity tag has been overexpressed. So you need to purify a protein from the E. coli. So you need to purify your protein from E. coli. So what step will you follow? So what you will follow affinity chromatography, obviously. So every option has affinity chromatography, but if you want to just purify a protein of your interest, so you need to have one more extra step after this affinity chromatography. So one more extra step will be there, affinity chromatography followed by ion exchange, which is followed by SEC. So basically three steps will be required. So you can read this and you can include this in your notes. For application requiring the purest protein, if you want any purest form of protein for any such application, you will start with the affinity chromatography. And after the affinity chromatography, so this will be your first step, affinity chromatography will be your first step. So affinity chromatography will be your go-to step, your first step for purifying a protein. And after that you can add some more extra step so that the protein will be very much pure. So for intermediate purification or polishing, so to remove the impurities or misfolded protein, you can also go to the ion exchange followed by the size exclusion chromatography. So C option is correct. So now what type of interaction happens in affinity chromatography between a protein and a ligand? Is it covalent? Is it non-covalent? Is it ionic or ionic plus covalent? So in between protein and a ligand, there is a non-covalent interaction. So the right answer for this question will be non-covalent interaction. So the type of interaction in AC, that is affinity chromatography, that is in between a protein and a ligand, is non-covalent interaction. But the interaction between a ligand and the matrix is a covalent type of interaction. Because अगर ligand properly matrix से bind नहीं होगा तो ligand will fall off, and अगर ligand ही fall off हो जाएगा तो protein of interest जो है वो किससे bind होगा? So you have to remember this clearly. यहाँ से दो क्वेश्चन निकल के आ सकता है. In between ligand and matrix will be covalent, and अगर बात करें protein and ligand की, that will be non-covalent. So that by altering pH you can alter. So by altering the pH you will get the illusion of your protein of interest. Now next question, in which you have to match this column 1 with this column 2. So here you have so many things mentioned here, so you have to match it correctly. So VOS and coworker, ribozymes, methanogens, resistant to dehydrating effect of hypertonic solution and therefore to plasmolysis. So this is matching with this extreme halophiles. It means they have resistance to what? So many dehydrating effect of hypertonic solution and therefore to plasmolysis. Extreme halophiles means they are capable of living even in high amount of salt concentration. So इसका मैचिंग किसके साथ हो रहा है? फोर्थ ऑप्शन का मैचिंग हो रहा है एक्सट्रीम हेलोफाइल्स के साथ. सो 4 का क्या हो रहा है? ए ऑप्शन हो रहा है. नाउ राइबोजाइम. सो एज वी ऑल नो राइबोजाइम इज अ कैटालिटिक राइबोन्यूक्लिक एसिड मॉलिक्यूल. सो सेकंड का ऑप्शन किसके साथ मैच हो रहा है? सी के साथ. नाउ the VOS and co-workers. So basically they have just characterized the prokaryotic organism into two separate domains. It means first का जो मैचिंग है वो हो रहा है 4 के साथ. फर्स्ट का D. So यहाँ पे एक बच गया methanogens which have a link in our tracing the evolutionary history. So B option मैच हो रहा है कि थर्ड का क्या होगा? थर्ड का होगा B. They have possible evolutionary ancestor of the nucleus of the eukaryotic cell. So this will be the answer. VOS and co-worker are responsible for separation of prokaryotic organism into two separate domains. Ribosome is a catalytic ribonucleic acid molecule. Methanogens have possible evolutionary ancestors of the nucleus of eukaryotic cell, and halophiles are resistant to dehydrating effect of hypertonic solution and therefore to plasmolysis. So through this also you can easily earn your 3 marks. Now let's solve the other question which is related to the matching, and this is from Animal Kingdom. If you have not read NCERT, so I will recommend that you should read it, because through that only you will be able to solve so many questions, because it will clear all your basic concept. So this is a very important question from Animal Kingdom. So in the column 1 you have animal name mentioned, and in the second column you have their characteristic. So you have to match the correct characteristic of each animal. So you should be aware about the starfish, that is the echinodermat, who are known for their tube feet. So थर्ड का जो मैचिंग है वो 4 के साथ हो रहा है. थर्ड का 4 किस में है? लेट अस फाइंड आउट. सो थर्ड का 2, सो दिस इज एलिमिनेटेड. थर्ड का 4 यानी ये ऑप्शन हो सकता है. थर्ड का 4 यहाँ भी है, एंड थर्ड का थर्ड यानी ये भी एलिमिनेट हो गया. यू हैव एलिमिनेटेड आउट 50% ऑफ द ऑप्शन, यानी कि आंसर क्या होगा? आंसर इधर सी होगा या फिर बी होगा. लेट्स फाइंड आउट. सो you must be knowing about the characteristic of sponges, which are also known as the porifera. So the sponge is a common name of porifera, phylum porifera. So these are known for having a collar cell in their structure, यानी 4 का 3. So 4 का 3 किस में है? C में है, यानी कि B भी eliminate हो गया. So the correct answer for this question is C. Now sea anemone. Sea anemone is a coelenterata, and so sea anemone is a coelenterata, and coelenterata are known for the diploblastic nature, यानी कि first का 2. Now blue fly. Blue fly is an insect. So blue fly will be matched with this 3 pair of joint leg. यानी correct answer इस question का क्या होगा? Option C. So this is all for today. I hope you liked it. Thank you so much for watching.